So we are going to go through the some case studies and some knowledge exchange in like nuclear isolation and things.
So I will show you some slides of what we obtained at the Genoscope for the client sample from Plants and Algae. When we start 5 years ago with the IC protocols, we worked a lot with the plants, and at the beginning there were many protocols that were available and we would like to compare the results we obtained for a brassicacea plant, Brassica rapa, and we compared the dovetail and arima and OC data. So the main differences between all of those protocols was the input, because in the dovetail we use only 300 milligrams, and it's without nuclear isolation. And for the arima protocol, it was 2 grams or 3 grams of material input, but we engage this material and we perform a nuclear isolation. There is a lot of version of the kits around the time, but the main difference between the arima kits version was the use of the cell-lytic sigma protocol to separate the nuclei, because in the past it was more manual version. So we published the results of the comparison in this protocol, and the main result for this comparison was for the Brassica rapa. We obtained slightly better results with the Omni-C protocol. So at the lab, in the past, we would prefer for plants the Omni-C protocol.
So you didn't count nuclei by the microscope?
No.
You just measured the weight?
Yeah. So we tested both for the arima protocol and the Omni-C protocol. We used it succeeded with a lot of species, a lot of different varieties of cabbage for the arima, but also for the Omni-C, we use it with success with banana, beech, and apricot, many things, but it requires a few adaptations. So regarding the nucleus digestion volumes, in the protocol, we have to perform serial dilutions, of the nucleus mix. So for the beginners, we have 3 conditions: nuclease A, B, and C. And we use the nuclease A for the nuclease B, and then the nuclease B for the nuclease C. We start the digestion, lyse the cell, quantifiabilize it, and verify that the chromatin is properly digested. And it's the expected profile. So we should obtain more than 50% of chromatin digested efficiency. Yes. The percentage of the fragment between 100 and 2,500 base pair should be more than 50%. So that is the expected profile. And the real life in the way. So here are the conditions, the 3 conditions that we have in the protocol. And here are many different plants we use. And as you can see, I think it's little, the profile small, but you can see of the 2 last line, all the profile most of the time I under digested. Well, at the lab, we had a new condition and we avoid the last one. For the new condition, if you see here, all the two last one are also under digested and the first one is okay. And in that case, the second one is better, more accurate than the first one. Another things that you can point out, sorry. So for the same sample, we perform two independent digestion and we obtain for the same condition two different profiles. So it depends on the day, the climate, how lucky you are, but it's not perfect. So when we received from the algae project our first algae, so it was brown algae, we said, let's try omnisci plants starting from 300 milligrams, and then the real life. For the latest QC, we also have under-digested chromatin. So you can see the under-digested profile here. So we decide to reduce the material input for the nucleus digestion. And Laurie split this sample, the last 100 milligrams in 3 tubes, and she applied the same nucleus volumes. And bad news, same player, play again, as she always says. So we reduce the material input from the nucleus digestion, we increase the volume of the nucleus, so the volume increase, and then, yes, we got it. So we should continue to the next step, and then the proximity ligation. The beads fully magnetize with the chromatin capture beads. So we contact the dovetail support. They advise us, they suggest us to reduce the lysates, which bind to the chromatin to minimize the impurity. So, as we discussed previously, we should perform 2 parallel PL reactions with only 200 milligram lysates and continue and combine the 2 reactions before library prep. But it chooses 2 reactions, and for 1 sample it's okay, but for a lot of samples it's very expensive. And the second option from Dovetail was to start with nucleides, so that's why we start with this new option. So for the phagocytation, the brown algae, we continue to isolate the nuclei. Dovetail advises to follow this protocol from Workman, and then treat the nuclei as mammalian cells. So we follow this protocol just in removing the miraculous by the strainer, cell strainer, because we used to do that in our protocols. And here are the results we obtain with the profile. So we start from 1 gram of material, and we obtain an over-digested profile, a second one, and an under-digested profile. So same player, play again. And then we start from the same input of the material, but we decrease the volume of the nucleus. And then, yes, we got it. And we should go to the next step.
Oh.
But yes, user cases, the real life. So the lysate input was 1 microgram. We obtained after PLG quantification 300 nanograms, and we going through the library quantification, and then we try. So same player, play again. And then we use two separate reaction lysates, PLD, and then yes, we got it. 200 nanogram for the library, so we were happy, but it's a very expensive genome, as you can see. So with the RECA project, hopefully we have only one type of brown alga, because it was a little bit expensive for us. Just a picture of what we tried with Amnesty and the ARIMA protocol with the nuclei isolation protocol from BOSS. So mainly for plants, it's okay, but when we reach the alga kingdom, it's more difficult. We succeed with Pausacium, as I showed, but with many try. We also succeed with Pelagomonas, that is a microalgae, but we fail with the Graclupia. The Graclupia, the same as you have. We send you. Osmundea is another red algae, and Colarpa is the green algae from the BGE project. And we also test Graclupia with the ARIMA protocol, but we never succeed. Just some picture of what we experienced with the weird things we have with the nuclei isolation, both with ARIMA and Amnesty protocol. So sometimes when we perform nuclei isolation, you can see weird thing. Like this, after the isolation, we have a little dot, white dot, then with a brown blurb and slime sometimes behind. So we don't know what we have to do with that. So, for example, for Graclupia, we just took the white dots, and we also took the brown slime, blurb, I don't know the term. And we try to perform the library, but we failed for both pellets. Okay. Sometimes when you perform the proximity ligation, you observe a bit clumping also. The beads slide down to the bottom of the tube, and it's difficult to pipette and to discard the supernatant. You also have the bead clumping, and you also observe colored supernatant, a lot of impurities. And during the IC library prep, we observe also wear profile, and also always with the ARIMA kit with plants, we observe this hole. Only with plant, with some invertebrate, we observe a hole, but not this artifact. It's very strange.
Karine, I have actually a question. So from administrative point of view, sometimes you need to calculate a priori before you start what will be the project cost.
Yeah.
So that the user will sort of have an estimate. Would it be safe for plant samples to include double?
Yes.
Should we just
Plant algae, because
plant algae and perhaps snails and stuff?
maybe
So basically everything invertebrate.
snails or worms. If you have crabs or seals, or it's bigger genomes, so sometimes we need to perform 2 IC libraries, or if it's the high repeat content genome, we need more IC library, and it's
insanely expensive.
Yeah, I was telling somebody that in our own lab, we will, if we know it's an algae, say, I'm sorry, that's And be a little more expensive because we know we're gonna have to try more.
Yeah.